Good afternoon, dear students. In the last presentation about mutation, we discussed about what are mutations, what is the rate and frequency of mutation, whether mutations are random or whether they are directed. Further, we also studied about the basic types of mutation, that is point mutation, and about base pair substitution. So based on molecular basis of gene mutation, we have two kinds of mutation: base pair substitution, also called as switches, and number two, frame shift mutation. Okay. And now, what is a base pair substitution? Today we are going to discuss this. We have already discussed, but we'll fastly take a recap. Now, base pair substitutions are the simplest kind of gene mutation where there is a change in a single base pair. Okay, actually there is a change in a single nucleotide in the DNA which changes the base pairing. So, substitution of a single base pair or change in a single base pair in a DNA leads to a type of mutation called as base pair substitution. Base pair substitution is actually of two types. Number one, transition type, and number two, transversion type. What is a transition kind of mutation? Where one purine is replaced by another purine or one pyrimidine is replaced by another pyrimidine. Like adenine is replaced by guanine or guanine is replaced by adenine. In case of pyrimidine replacement, cytosine is replaced by thymine or thymine is replaced by cytosine. What are transversion mutation? A transversion is a kind of mutation in which a purine is replaced by a pyrimidine or vice versa. Pyrimidine is replaced by a purine. Like adenine can be replaced by cytosine or thymine, guanine can be replaced by cytosine and thymine. Similarly, a pyrimidine like cytosine or thymine can be replaced by either guanine or adenine. So, the number of possible transversion is double the number of possible transitions. Okay, one base can undergo only one kind of transition, but one base can undergo two kind of transversion. Okay, but actually in vivo, in living organism, transitions are more common as compared to transversion. Okay. So let us discuss about various kind of mutations that occur because of base pair substitution. That is because of transition or sometimes transversion. So base pair substitution. Okay, replacement of one base pair by another base pair leads to four different kinds of mutation. Number one, missense mutation. Number two, nonsense mutation. Number three, silent or neutral mutation. And number four, mutation in termination codon. Missense mutation we have already discussed, but we'll discuss it again. What is a missense mutation? It is a kind of mutation in which there is change in only one amino acid in the protein chain. Okay, so missense mutation is caused because of base pair substitution, wherein a base present in a DNA is replaced by another base, because of which the triplet codon in the mRNA also changes. So substitution of one base in the gene changes or alters the codon in the mRNA. Okay, this results in incorporation of a different amino acid in the protein chain as compared to the normal. So missense mutations are those wherein there is replacement of one amino acid by another in the protein chain. So if you look at the normal protein chain and the one because of missense mutation, you will find difference in only one amino acid. Okay. So if we take an example as shown here, a triplet shown here, see, you can see TCA here, okay, TCA, and here the one of the base C has been replaced by thymine. Okay, so it becomes TTA. Now, as the mutation, as the DNA replication process proceeds, what happens? In the normal course, the mRNA contains the codon UCA, which codes for serine amino acid. So in the normal chain, there is serine. But because of mutation, because of base pair substitution, the codon of the mRNA gets altered, gets changed. That means UCA is converted into UUA. because of which instead of serine, there is now leucine. So there is change in one amino acid in the whole protein chain because of mutation. The classical example of base pair substitution leading to missense mutation. Rather, you can say the classical example of missense mutation is sickle cell anemia disease. Okay, sickle cell anemia occurs as a result of nucleotide substitution. One nucleotide substitution. And of course, because it is a kind of sickle cell anemia, it occurs in hemoglobin gene. So what is a missense mutation? It is a base pair substitution which results in substitution of one amino acid, one different amino acid in the protein chain. Missense mutation substitute one amino acid for another. Okay, some missense mutations have very large effects. That means sometimes the effect of missense mutation is so deleterious, it is so harmful that it can cause a severe problem, disease like sickle cell anemia. Okay, at times it may not be so harmful, but at times missense mutation can be very, very harmful. Okay, say for example, sickle cell anemia. What happens in sickle cell anemia? In case of beta globin gene, okay, there is change in the beta globin gene. Say normal hemoglobin is represented by HbA and sickle cell hemoglobin, as we have shown here, is represented by HbS. Now, what happens in sickle cell hemoglobin? Sickle cell hemoglobin, in case of sickle cell anemia, one of the codon from the N-terminal end, one of the codon is GAG. Okay, if you go from N-terminal end towards C-terminal end, this GAG codons for glutamine. It codes for glutamic acid in the normal protein chain. Okay, but because of mutation, the GAG codon is converted into GUG codon. Now, the GUG codon codes for valine, which is a hydrophobic amino acid. And when the protein structure folds, gets stabilized by tertiary and quaternary stabilizing bonds, what happens? Valine, which is a hydrophobic amino acid, comes on the surface when it is exposed to water. Okay, now on the surface of the globin molecule and gets exposed to water. And under low oxygen condition, valence affinity for hydrophobic environment causes the hemoglobin to crystallize. Rather, you can say that because of a missense mutation taking place in one of the, you can say nucleotide base pair, changes, here we have shown, changes the codon GAG into, 1 minute. It changes the codon GAG into GUG. This is the GAG codon. It changes to GUG codon. Because it has changed into GUG codon, instead of normal chain contains glutamic acid, and in the abnormal chain there is valine. So glutamic acid is replaced by valine. Okay. Now normal hemoglobin chain contains 2 alpha chain and 2 beta chain, and this change happens in the beta globin chain. Right. Normally, the RBCs are disc shaped. They have they can transport oxygen to different parts of the body. Okay. They contain hemoglobin pigment, which transports oxygen to different parts of the body. In case of sickle cell anemia, where glutamic acid is replaced by valine, the RBCs are disc shaped, sickle shaped. They have low oxygen carrying capacity, and as soon as they transport oxygen, they get degenerated. The life of the RBCs is also less. So a small change in a nucleotide changes a single or alters a single triplet codon, which changes a single amino acid. But change in a single amino acid can lead to a drastic effect like sickle cell anemia, which is transmitted from one generation to another generation. And it is a serious deleterious effect that is caused by missense mutation. Okay. So another kind of mutation is nonsense mutation. Now, if you look at nonsense mutation, what is a nonsense mutation? What is a nonsense mutation? It changes a sense codon into a nonsense codon. Okay. During mutation, some changes are brought about. There is base pair substitution in the gene. Because of, as you can see here, TCA changes to TGA, base pair substitution. One of the base is changed. So instead of CG pairing, okay, C changes to G here. Now what happens? Okay, because of base pair substitution in the gene, it changes the codon in the mRNA. Okay, so the normal codon in the mRNA was UCA. Now it has been converted into UGA. UCA codes for serine amino acid in the protein chain, whereas UGA is a stop codon. Okay, that is what is nonsense mutation. Nonsense mutations are those in which there occurs a mutation which changes a sense codon into a nonsense codon. See, there are 64 codons. Out of the 64 codons, 61 codons code for one or the other amino acid. Okay, they are sense codons. The three codons UAA, UGA, and UAG are called as chain termination codon in prokaryotic organism, or they are called as nonsense codons. So any mutation which changes a sense codon into a nonsense codon are called as nonsense mutation. Now, because a sense codon has been converted here into a nonsense codon, the protein polypeptide chain synthesis abruptly stops. Okay. So initially there was UCA in the chain, it codes for serine and the protein synthesis continues. But because of mutation, UCA has been converted into UGA. UGA is a stop codon. Okay, tRNA does not contain any anticodon for it, and hence the protein synthesis abruptly stops. So what happens because of this? Premature chain termination. Because of premature chain termination, what will happen? The protein formed will be shorter in length. And because the protein formed is shorter in length, it may be partially or totally non-functional. Okay, or it may have a long-lasting drastic effect also. Okay, most of the nonsense mutations are, you can say, not so harmful, but often nonsense mutation result in completely non-functional protein because the protein formed is shorter in length. Why? Because there is premature chain termination. Say for example, hemoglobin, a beta chain of hemoglobin contains 146 amino acid. If mutation occurs on the codon number 145, UAU, which codes for tyrosine, and if UAU is converted into UAA, which is a stop codon, what will happen? The final protein will contain only 143 amino acids, right? So the chain will be shorter, and that shorter chain may have a drastic effect or may not have the protein chain may be functional or non-functional. Okay, if such a change happens in the hemoglobin beta hemoglobin chain, then it results in thick blood, formation of thick blood because of overproduction of RBCs, which can lead to abnormal bleeding or clotting. Okay. Third type of mutation because of base pair substitution is silent mutations or neutral mutations. Now what happens? There are 61 sense codons, but there are only 20 amino acids which are carried by 20 RNAs. Okay, that means more than one codon code for the same amino acid. More than one codon specify the same amino acid. Like 2 codons may be adding the same, like 4 codons or 6 codons may be adding the same amino acid, like leucine or isoleucine. Is that clear? That is called as the code is degenerate. We say the code is degenerate. Because of the degeneracy of the code, more than one codon code for the same amino acid. So silent mutations are those mutations in which, though the mutation changes the gene, though the mutation changes or alters the codon of the mRNA, but the amino acid coded or the amino acid specified by the changed codon is similar to the one which is present in the normal case. Okay, so silent mutation alters the gene, it alters the codon in the mRNA, but because of degeneracy of the code, same amino acid is specified. Okay, so though mutation has taken place, mutation has changed the gene sequence, mutation has changed the codon of the mRNA, but the amino acid present in the protein chain is similar. There is no change in the amino acid sequence. So the protein formed is totally functional. It does not have any effect on the phenotype or functionality of the protein. Such mutations in which, though mutation has taken place, but there is no effect on the function of the protein are called as silent or neutral mutation. Here you can see that The codon is UCA. Normal codon is UCA. Because of mutation, it has changed to UCG. But both UCA and UCG code for serine. So the same amino acid is present in the protein chain, right? So base pair substitution, which result in same amino acid, the protein is not altered, are called as silent mutations. Okay. Like for example, AUU, if it is changed into AUC, both of them code for isoleucine. So there will not be any change in the protein chain. This is called as silent or neutral mutation. The fourth type of mutation which occur because of base pair substitution are mutation in termination codon. These mutations are reverse of nonsense mutation. What happens in a nonsense mutation? Sense codon is converted into a nonsense or termination codon. And what happens in this kind of mutation? Sense mutation. That means a nonsense codon is converted into a sense codon. Any mutation which substitutes one base in the gene by another leads to converting a nonsense codon into a sense codon. Here, a stop codon or a nonsense codon is converted into a sense codon, which codes for an amino acid. And hence, the protein synthesis continues even after chain termination to be taken place. Like there is a stop codon in the chain and chain has to terminate. But because the stop codon has been converted into a sense codon, the protein synthesis will continue further. Okay, but most of the times there are 3 stop codons. Actually, in the end, there are 3 stop codons, maybe after a few codons, right? So after synthesis of few more amino acid, the protein chain synthesis will stop. Okay, but sometimes, okay, but what happens here in if mutation take place in nonsense codon and it is converted into a sense codon, the resulting protein may be having a greater length. Okay, the amino acid present in the protein will be more as compared to the normal one. So the resulting protein gains additional amino acid and it is longer as compared to the normal one. So most of the time it may not be harmful because the extra amino acid may not be taking part in the stabilization of the structure of the protein and it will not affect the protein. But sometimes these extra amino acids or the the protein chain having extra larger length may have a drastic effect. Like say for example, in case of thalassemia, it's a severe kind of anemia. Okay, wherein the alpha globin gene, which is normally 141 amino acid long, okay, here mutation in the termination codon UAA converts it into CAA, that is glutamine. So because UAA is converted into CAA, the amino acid synthesis will continue and the resulting protein will have 31 additional amino acid till it reaches the next stop codon. So that may have a very deleterious effect, like it may cause thalassemia. So be it missense mutation or nonsense mutation or mutation in termination codon, well, sometimes it may not be affecting the phenotype of the organism or it may not have a drastic or a harmful effect, but sometimes it can have a very drastic deleterious effect on the phenotype of an organism. So these are the 4 different types of mutation resulting because of base pair substitution. And in another lecture, we will discuss about the frame shift mutation.